PhD in metabolic biology from Columbia University, followed by a postdoc in the Laboratory of Biochemical Genetics and Metabolism at the Rockefeller University, where she now serves.
Have real conversations about this stuff. This is and so we George Beadle mentioned in the quote that Liz discussed about how we've been doing genetic engineering for eons, since the dawn of human civilization. And that was just by selective breeding. You know, people would breed and cross wheat crops that seemed to provide bigger yields. People would selectively breed cattle so that they can increase yields for whatever dairy or meat or whatever. And so genetic engineering has actually been something that's been done for a really long time. We just never called it that. But what we're talking about here is something that goes a little bit above and beyond sort of the selective breeding. We're talking about actually going in and manipulating the genome, manipulating the genetic code that's inside of cells. And so this is really a new frontier, a newish frontier. We've been manipulating genomes for a little while, but the technologies have advanced so far that it's time to start talking about it on more broad levels. Okay, so let's go into a little bit of what is genome editing anyway. Does anybody have an idea? People have been doing it in model organisms for a really long time. You know, I have done it and a certain aspect of biology because this gene from the jellyfish glows really bright. And if I attach it to something inside of a cell, I can track it inside the cell and understand, like, where does it go? How does it behave? So this is definitely something that's been done. Yeah.
Is Roundup Ready a good example of editing?
So yes. So Roundup Ready is basically taking a crop where you insert a resistance gene into it, and so you can spray these pesticides, and it would kill all the weeds and allow the crop to still sustain itself. And so that's a controversial thing because in theory, it's a very good thing to be able to have healthy crops. But what the business model, I think, is maybe beyond the scope of this discussion, but it's definitely an important discussion nonetheless. Yeah. Okay. All right. So I like to kind of draw an analogy. The way we're looking at genome editing is sort of the way, does anybody use Microsoft Word or Google Docs? You know, you can go in and you can edit whatever. So genome editing is essentially word processing for your DNA or anybody's DNA, where you can, like, you know, change the meaning of a sentence or maybe add to the sentence or do a number of modifications to those sentences. So let's just, for really quick, do this. All right, so we have two sentences. The cat jumped off the chair, very clear meaning, right? Or the dog ran around the park, very clear meaning. And when you're doing word processing, and similar to the DNA, you can do things where you delete something, right? So now it becomes the cat jumped or the dog ran. Okay, so this changes the nature of that sentence. It changes the meaning of that sentence. We can do that with DNA. We can add something to it, right? So the cat jumped off the chair into my lap. Now it sort of makes it more specific. And that's kind of when we add something to it. This is kind of what we're talking about when I said I took a gene from a jellyfish and attached it to something inside of a cell and followed it, right? Or the dog ran around the park until it was tired. So we're adding something to the sentence. And similarly, we can cut and paste and change it all together. So now it becomes the dog jumped off the chair or the cat ran around the park. And this is another functionality that is afforded to us by genomic editing. Okay, so basically genome editing is essentially word processing for DNA. And our DNA is essentially just code. Right? It's A, T, C, and G. And the order of that code essentially makes us who we are. It differentiates us from a broccoli or a tree or a dog, right? So, and that change is actually very, very, very small. But, you know, majority of us share a lot of DNA, and that's the beauty of this sort of idea of doing science on genetics and model organisms. Okay, so while genome editing technology has been around for a really long time, the one that's getting the most press and the one that you might have seen in newspapers is something called CRISPR. Now, CRISPR is essentially an acronym. It stands for Cluster, getting really excited about its uses for humans. And so now we have this tested. don't worry. So the top, that sort of red kidney bean shaped kind of thing, is part of the machinery. That yellow genetic piece of information is called a guide RNA. It's essentially how you trick it. This is the GPS. So this is the sequence of DNA, this is a sequence of nucleotides, or A, T, C's, and G's, that you can program to give the machinery itself how to find the DNA that you want to alter in the cell. So this is basically giving it the information to go toward that DNA. And so on the left you see you can use it to get rid of some code, right? So you are disrupting a gene or mutating a gene, maybe that's very useful in science. Or on the right, you're inserting something. This is where, for instance, I would insert that gene from a jellyfish into another piece of DNA. This is going to be the most complicated slide that we have, so yeah.
Do you have to have the full genome for the bacteria that you're trying to trick so you know where?
So you're actually only taking out the machinery from the bacteria. You don't need the genome of the entire genome of the bacteria. You're just doing the machinery. And you have to know the sequence that you want to alter. Yeah. But you don't need to know everything else. Okay. Any other questions before we move forward? So I'm just trying to give you guys a primer, but I do really want to have a discussion as we move forward. Okay. So the potential applications of CRISPR are just extraordinary. They're so exciting. Okay, for instance, we can use it in agriculture to create crops. I mean, think about it. By 2050, the human population will be reaching 9 billion people. How are we going to make sure that we can feed people? How are we going to make sure that we can just sustain the basic rights of life, right? And so agriculture, this is a huge application for agriculture. We can generate crops that are maybe more robust, create more food. Another thing that we can do, and what has been done very recently actually, is to genetically alter some of the dairy cattle so that they are born without horns. Now this is actually an excellent thing for animal welfare because the horns do pose a problem for animal safety and handler safety. And imagine you can also take yeast, for instance, and program it to produce really, really healthy, for instance, cooking oils. You know, cooking oils that are high on saturated fats that people could use in their salad dressings. That could also be a really interesting way to use it. Another interesting way that you can use it is by the creation of pharmaceuticals. So you can alter different types of plants or different types of cellular organisms to basically generate small molecules that can be used as drugs for people when they're ill, or maybe produce vaccines or something like that. So there's a lot of really interesting ways that you can use it. And really the potential applications of CRISPR are utterly endless. They really are. It's just what can you think of? Because that's what you can do, right? But does this mean that we should not draw a line? And this is the question I want to focus on mostly for today. Okay? So let's talk about CRISPR and its uses on the human genome. Okay. But before we do, I want to talk about the difference of cell types in humans, or in basically lots of animals, but we'll talk specifically on humans. So there's an idea of germline cells versus somatic cells. Every cell in our body has DNA, right? And some of the DNA would be, in specific cell types, would be passed down to certain generations. And those are our reproductive cells, right? And so these are called germline cells. These are the cells that are carrying the information as parents have children and their children have their children. And that's why, for instance, I maintain a gene for like blue eye color, and or you might maintain a gene for your height or, some other genetic predisposition. And so these are a really, this is the question I want you to kind of think about in a little while, is the difference between germline cells and somatic cells. Somatic cells, on the other hand, are cells that are important for our body systems, but are not the cells that we're, passing down to the next generation. So cells that make up our skin, cells that make up our liver, cells that make up our blood cells, cells that make up our muscles, those are somatic cells. So that's a difference between somatic and germline. So when we think about genome editing, we have to think about genome editing of somatic cells versus genome editing of germline cells. Okay, so think about that moving forward. Okay, so now I'm going to talk a little bit about the 4 buckets of using CRISPRs, genome editing on humans. And I'm going to do it from, I think, what we see as sort of least problematic to maybe most problematic. Okay, so we'll start with the least problematic. All right, so one of the most versatile ways that we can use CRISPR for understanding human biology is in just simply basic science research. So we have cells in a dish, we add CRISPR to modify some aspect of their genome, and then we just watch and see what happens. And that allows us to basically understand pathways or processes, or for instance, you can take a cancer cell line, you can use CRISPR to edit some sort of gene in that cancer cell, and then see, are those cells still cancerous, right? So this is a really useful application of CRISPR in basic science research. You can use it in flies, you can use it in yeast, you can use it in other bacteria, not just human cells, any cell type. So and this actually is being done, every laboratory that performs genetic experiments at Rockefeller, they are essentially using CRISPR, so this is a very widely used tool. Okay. So the second really interesting way that we can use CRISPR is to treat disease in humans. So essentially what you could do is take out some of the diseased cells in a human, modify its genome in a way that you think will remove that disease or reduce that disease and put them back into the human. So this could be really valuable, for instance, in cancer, or perhaps HIV. Or there's actually some clinical studies going on right now. I believe there's a clinical study that was just approved where there are 18 patients who are getting a tumor cells removed. Their cells are being modified, and they're going to be put back, and we're going to see what the effectiveness is on fighting that cancer. There was also an example of a young teenager in France who had sickle cell anemia. And what they did was they removed a large portion of cells from her bone marrow. They modified those using CRISPR to produce more functional hemoglobin. And at the same time that they're doing that, they're basically irradiating her body so that all of her bone marrow cells are getting killed, right? And then they put the modified bone marrow cells back into her, and what happens? It's been 2 years, as it turns out, she has not needed a blood transfusion. And she is showing a 50% increase in hemoglobin functionality. So this was a report that was just sort of released. And I know that people are looking into it for HIV, leukemia, other types of aspects of it. And this is sort of like more and more clinical studies are undoubtedly going to be entering into the conversation in this way. So I think CRISPR here, it's a no-brainer for me that this is a really interesting and valuable way to use CRISPR. Okay, so here's where it gets dicey in my mind. Okay, so now we can use CRISPR for the prevention of heritable diseases. Remember we talked about germline versus somatic. So here this is a somatic cell, right? This is not going to be passed down to the next generation. Here, this is a germline cell. This has the capacity to be passed down to the next generation. This is heritable, okay? All right, so basically here, say for instance, you are a carrier for a disease. Let's talk about perhaps Huntington's disease, okay? So Huntington's disease is a dominant genetic disorder that most people don't know they have until they are like 35 or 40, right? So generally what happens with Huntington's is that people have children before they go, and so Huntington's disease is continually passed down, okay? Now imagine now that people are doing more newborn screening, they're doing more genetic screening. You can identify this very early on, and you can understand if you are a carrier or not. And if you are a carrier, because it is a dominant disorder, you only need one copy for this disease to come to fruition, right? So imagine being a carrier, as I am a mother, right? If I knew that I was a carrier for Huntington's disease and I know that I wanted to have children, what I might do is then proceed with some sort of, take my eggs out, modify my egg, get rid of that Huntington's mutation, and then stick it back in and have a baby so that my kid is guaranteed not to be sick. I would want to do that. But where do you but how do you categorize? This should only be done if there's no alternative, right? There's no other way to do it. And how do you establish that, and where do you draw the line? What disease is considered yes, no alternative, and what disease is like, well, sorry, you're gonna have to live with that, right? So where do we draw that line? And that's a fuzzy line. And then here's the fourth bucket, right? Genome editing for enhancement. Who has seen Gattaca? Do you guys know that movie? So this is a movie from, I don't remember, I think maybe the late 90s, early 2000s, where basically there was two societies, sort of the working class society and then the elite society. And the elite society had their genome, it had a very specific genome, and only they could be sort of the people that make decisions for humans. And it's a reality that could very much be, this is not fiction anymore, right? And so is it okay for someone to say, you know what, I want to have a baby. When we're talking about germline editing here in this, I want to have a baby that will have a high IQ, will be 6'2", has really athletic build, just basically getting your kid to look and feel and just be, what society considers as ideal, right? Is this okay? Okay, so as we discussed, I think it's important that we have to go beyond pure science, right? Talking about science for the sake of objective science is one thing, but talking about science for its application in society is a whole different ballgame. We have to understand the cultural identities and the philosophical identities of all of the communities that we want to help in these cases. We also have to take into consideration, finances and socioeconomics. Like this stuff, if you were to do this on your germline, so if I wanted to take my eggs out and do this thing, this is undoubtedly going to cost thousands, if not millions of dollars. I don't have those funds, but maybe someone else does, especially the type of person that might purchase a trip around the moon. They can do those things. And so then you're drawing at who has access and who does not. And these are questions that we really need to tackle. So if you're going to have this be available to humans, it's either all or none, right? Or not. And these are questions that we have to talk about. So these are the questions I want to pose to you guys. Before we go, actually, does anybody have any questions on the sort of scientific aspects? Yeah.
It's going back to a slightly earlier stage.
Sure.
I just wanted to ask you, does GMO use CRISPR?
So GMOs are, yes, they can, that's basically taking what we know as one gene from something and putting it into something else. This is what we're doing a lot for crops, and you mentioned the Roundup Ready. And yes, so CRISPR is one of the tools that you can use to create a GMO food product.
And just to follow up, when was CRISPR developed, and was GMO being done before CRISPR was developed?
GMOs have been being performed for, I think, like at least 30 years. Has anybody, do you guys eat corn? There's very few corn. You are guaranteed to be eating corn, GMO corn. We have very few corn available that are not GMOs. Also similarly with soybeans. It's just the fungus, the fungal outbreaks that kill those crops are very, very robust. And so without the GMO, we would have essentially no corn in majority of situations. So, yeah. And so CRISPRs, so as I had mentioned, CRISPRs were discovered, I think, within the last couple of decades, maybe 10 or 20 years, at least they knew that these sequences were present, but nobody had sort of thought about it for its application until 2011. So this is very fresh. Yeah.
I'm not sure that it's appropriate, but when I buy some lettuce, for example, that was grown hydroponically, it goes bad in 2 days. If I buy some lettuce that's grown in the soil, that stays for a week or two and it's still good.
Yeah, so that could be a difference in lettuce species, it could be a difference in how long the hydroponic was chilling out on the shelf before it got into your fridge. I mean, there's a lot of things to consider on that.
For me, it's where it was grown.
So that's not a genetic, that's an environmental factor, so that is not genetic. But I will say, people have been using CRISPRs to create, for instance, mushrooms that do not turn like brown, or actually also apples that don't turn brown. So people are doing it for aesthetics. I have no problem eating a brown apple, but some people might, so that sort of gets at that.
It's an
Yeah,
oxidation.
yeah, it's oxidation.
I'm just wondering, is there any information on the people who have gotten the treatment through CRISPR? Are there any adverse events that they've experienced?
So far, there have been few people that have received these types of treatments. The report on the young teenager from France with mesothelioma just came out within the last couple of days, and the report suggests that benefits. However, it does not mean there's still like off-top. So CRISPR on human embryos has occurred in China and in the UK. In China, they were not able to do it successfully, even though they tried. And that's because I believe that there are some off-target effects that are impacting the experiments. But so far I have not, and also there are regulations, I believe it's an international regulation where you are not allowed to keep a human embryo alive beyond 14 days. So doing that 14-day window is actually really valuable in understanding. We don't know exactly what happens during development of an embryo, and so this is for stem cell researchers, this is a very important set of experiments in helping them understand just what are those processes during development, and that could be a beneficial application later on for people that might have trouble with pregnancy or something like that. But right now, international sort of agreement is 14 days is the maximum that you are allowed to keep something like that alive in a test tube.
Oh, okay.
Any other questions before we get into these other philosophical questions? Okay.
Okay, well,
So I would like to hear from you. I want you to think about, where would you draw the line? I'd also like you to think about who should be the ones to decide. Yes, scientists are the ones that are pioneering this stuff, but they are certainly not the only people who should have a say in what we do with this, right? How do you ensure equivalent international policy? As I just mentioned, both the UK and China are okay with doing CRISPR editing on human embryos. But in the United States, a report was recently published by the National Academies Press where the people who wrote that report felt that we should be able in the United States to proceed with editing human embryos using CRISPR, following these guidelines of no more than 14 days allowed in to go beyond that. But they are saying that it should be done, but proceeding with caution. So this is the recommendation of this panel. But how do you make sure everybody's going to follow stuff like that, right? How should access to this technology be regulated? As I had mentioned, if I wanted to edit my own genome for future, babies, I'd have to spend a huge chunk of change to do this. This is not covered by insurance, right? So how do we make sure that anybody who wants to do this has access to that versus only the people with means? And lastly, how would you draw the distinction between genetic disease, like helping to fight a genetic disorder, versus simple enhancement, right? So for instance, another example I want to share with you is that there is a genetic underlying basis for instance, muscular dystrophy. And you could essentially use CRISPR in theory to improve muscle tone in the child, the future child. Muscular dystrophy, for those cases, that could be really, really valuable and effective. But where do you draw the line for instance, improving muscle tone? Is muscular dystrophy the only category that you would think would be appropriate for this, or are there more? And how do you make those decisions? And so I'd love to hear from all of you on any aspect of these questions, or maybe a question that is not up here. So where do you feel comfortable using CRISPR? You feel comfortable using CRISPR, yeah.
So I'm going to start right at the extreme end of the spectrum and say I would draw the line to across, or to prevent any kind of genetic manipulation, because essentially what genetic
So even here,
yes, because I'm just going to take this position just to explore the extreme.
Sure.
Because the essentially what you're doing here is you're replacing natural selection. So this opens the door to.
one might be able to say that you're speeding up natural selection.
But it's not natural.
But you're using DNA, which is a naturally occurring phenomenon. I'm pushing you hard back, you know?
Absolutely. But just to finish my argument, I think
Sure.
that what's happening here is that human beings who are provenly fallible, we developed the nuclear
Manhattan Project.
Project, and we clearly don't know how to handle it.
No, we do not.
Thank you. Thank you for that.
Yeah.
So now we have a similar technology which can be used for great good, but also for great evil. And is it going to get? Are we evolved enough as a species, as a civilization, to be able to handle it? So I would say, draw the line, and let's explore if we're ready.
Yeah, and I think that's definitely a stance that is not a minority stance. I think there are people who feel uncomfortable with that. I do want to report out though that genetic manipulation has already been happening for decades. With the there was this sort of a discovery many decades ago, and maybe in the 60s, of restriction endonucleases, where it allows you to cut and paste things into DNA. However, the efficacy and sort of the preciseness of that was not very good. So you would just cut and paste wherever and plug it in and hope that you got the thing that you wanted, kind of thing. So there's a spectrum of that. But with CRISPRs now, it's much more precise, and so it's almost like a better way to do it because you're limiting off target and just random stuff. Right.
One thing that I see that is a really large benefit is that so many people suffer with rare diseases
Yes.
that there's no financial incentive
None.
to cure
Right.
rare diseases. So I have a niece who has a congenital genetic illness.
Sure.
And it wasn't diagnosed until she was 6 months old when she was having seizures, and they don't know how long she may have been having seizures for.
Right.
And she has, it's basically the reverse of diabetes, where her body can't regulate insulin.
Sugar,
So
yeah,
her blood sugar is always dropping, no matter
Sure.
how much food she eats. And there's very few treatments for that, and there's, like I said, not a lot of incentive. And there
Which
are,
I should mention that's changing. There are a lot of companies that we
That are
just
focusing on
celebrated Rare Disease Day
Right.
last week, so there are things.
right,
Yeah.
so that might be changing, but this is something that I can see that would be a way of being able to, I think the old treatment for hyperinsulinism was like cutting out 90% of the pancreas. And
Right.
Seeing how
Right.
patients did.
And that's an invasive procedure.
Sure, so if you have something that is less invasive and more precise, like you just mentioned, than older genetic treatments, if you knew exactly what part of potentially the
Yeah.
germline to target. If my
In
cousin
that
wanted
case, that you wouldn't be able to treat a genetic disorder in a human that's already in existence through
That
germline
has to be
modification. You would have to do it through
somatic.
somatic modification.
But if you knew that it was a genetic illness that you might pass down to your children.
Yes.
And you could target just that.
Yeah. As a mother, I'm speaking strictly from a mother, not as a scientist, I would do anything.
Yes.
Anything. I would break
Children
the
will
law
lifetime.
to save my children. Anything. There is nothing I would not do to make sure my children were okay. And so, yeah, I do favor that type of.
And if it could be eventually a more financially feasible way of targeting rare genetic illnesses.
Yeah.
That improve quality of life.
Absolutely. I agree with that as well.
As a mother, I agree. I would do anything for my children. But if CRISPR is going to happen regardless, then maybe we should concentrate more on controlling it. Do you see that occurring regardless of our opinion?
So there has been a summit to discuss the future of using genome editing, which includes CRISPR. And in that meeting, it was absolutely clinicians and scientists, but there were also ethicists, religious leaders, economists, policymakers, basically a lot of people from all different aspects of just human life coming together to just discuss the status of where the science is and what to do next. So those conversations are being had. I can't say that I fully understand the impact of those opinions and how they would shape the policy coming forward, but I do know that they are happening and they are open. But that does not mean that we shouldn't be, voicing our opinions and discussing this type of information and potential policy with our legislators moving forward. Yeah.
Is there a group or groups that are working together to try to influence the policy in reference to?
Undoubtedly, I think, I can't say that I'm equipped at this point in time to list out who those groups are, but I know, for instance, I have some colleagues at Harvard who are involved in basically promoting genetic literacy to the general population, so I know that they are very much involved, and they do it from ethical standpoints, not just sort of the pure scientific standpoints. So I do feel that these conversations are being had, and I think as we, and my goal, sort of what I want to see, is just to raise awareness that these are things people are talking about in scientific communities or medical communities, and to encourage people who are not scientists and clinicians to get involved in these conversations and to voice where you stand here, because I think it's really important. It's too long that we've kept these sort of scientific ideas in sort of these compartmentalized buckets that science impacts society at every single level. And I believe that we should have more open channels to facilitate not just the education of non-scientist populations, but also just to understand where people are at, what is relevant to them, what is important to them, and where do they feel comfortable with this type of technology moving forward. Yeah.
Is there anyone who's approaching this entire concept, this is kind of a weird question, not from a human-centric position? So, I mean, I think of Jane Goodall, who was like, yeah, if I could do anything to help the earth right now, and I could do it without causing any human suffering, I would get rid of lots of people, because there's way too many of us, and
9 billion is definitely beyond the carrying capacity of planet Earth, at least the calculated carrying capacity.
right, and it just seems like, I mean, of course, I think probably all species and just maybe part of nature is like, you try to make sure you survive, and so you do stuff to make sure that the other things that are like you survive, but also, like, we're killing everyone and thing to make sure that we survive. So I'm just wondering if there's any movement within the scientific community to say, how can we use this not just for human benefit? I understand the agricultural stuff, but the agricultural stuff is
benefits humans.
Right. It's not like,
That's a hard question. I mean, I can imagine maybe, for instance, and I'm just going, I'm making this up right now just as one potential application that has nothing to do with humans, but like, for instance, I know there's a lot of coral bleaching and stuff happening right now. Imagine being able to use CRISPR to improve the ability of coral to sustain all of the environmental pressures that are, I mean, that I might have like a secondary impact on humans because we get to scuba dive and see it, it doesn't like destroy tourism, but that could potentially help ocean health, which could then potentially help humans later on by ensuring food security or something like that, right? So I'm not sure how people are thinking about it in terms of that's a great question, but I know that people are typically, it's hard to get stuff funded unless you have an angle that benefits humans. So that's just how science funding goes. People want to know why are their tax dollars being used to understand like beak size in a chicken. I don't know. I'm just making stuff up here. When there are people with legitimate diseases that need help, right? So, yeah.
onto a very deep ethical question that I never thought about before, a couple of weeks or months ago, which is that, you know, as our medical processes and science has expanded and research has gone further, we're able to now, you know, mothers can have babies at 20-something weeks and they all survive, whereas 100 years ago that wasn't true.
Right.
And so all these babies that we don't know what they have and, you know, what happened that they were born so early, what are they passing down genetically? And the fact that we're able to have people thrive with certain conditions who weren't able to before, and they're having more children than they were 50, 100 years ago. What is happening to our whole gene pool
The gene
as a
pool, yeah.
total? I mean, it's almost like the opposite of survival of the fittest because our technology has advanced. So it's this difficult thing of, well, we want to save and make everyone have as good of a life as possible, but what is it doing to everyone as a
Right, so
whole?
what that I think is the angle of placing individual above community, right? That's a good point, and it's hard to really get into a conversation like that without coming across as insensitive sometimes, you know, and but it's true. I'm not sure. People call it like playing God, you know. Where are we on that? And that's it's hard to think about. Yeah.
So I remember seeing a picture of kittens that are cloned, and they all look pretty different. So when it comes to gene editing, how do we know that it's just having the gene will ensure that that trait or feature is activated?
Yeah. So that is the million-dollar question, right? So the whole idea with the experiments that happened in China where they did the genome editing of the human embryo, it didn't take because of presumably off-target effects, and so they're trying to figure out what does that mean, and I mean we don't know what we don't know, and for me as a citizen and not as a scientist, I love we don't know what we don't know because I just want to go deeper and deeper and deeper, right? But as a citizen, non-scientist, I'm too scared by that, you know? So I'm constantly like grappling with that sort of balance of what happens if, and we're also learning that it's not just us in our bodies, it's bacteria, it's yeast, and there's this intricate network of genomes that are really working together over the course of evolution. I mean, we have never been alone as humans or as Neanderthals or, you know, whoever, Australopithecus, whoever came before us. Every single evolutionary organism has had another organism that they were in a relationship with. And so if you impact a gene in one organism that's heritable over time, or multiple genes, how does that impact the relationship with that other organism, right? And so we know that we need all that bacteria for our gut health. We know that we need all that bacteria for, you know, our skin to function properly. I mean, I'm not saying that it will impact it, but I'm also saying we don't know if it will impact it, you know, and so those are things that are definitely worth considering and studying before I think humans are put forth as genetically engineered organisms. Yes?
More of a statement, but suppose that we do have trials of editing human
Like germinal
embryos.
cells. Yeah.
Yes. Like, what about the legal ramification? Someone who was born to these experiments and now they're not getting covered by insurance.
Pre-existing condition.
Yeah.
Yeah, so that raises another important issue and one that I am very passionate about, and that's one of consent. Okay, so yes, it seems like it's a good idea to edit this kid because they won't have this X disease, but we never ask permission to that child to be able to do that thing, right? And so, what does that mean? What does that mean to you? Like, do you think it's okay to genetically engineer a child and then have them be born and just be like, surprise, we made your DNA the way we wanted it instead of the way it was supposed to be? Like, what do you think about that? Does anybody feel that's okay?
I think it's about genetic resolve a genetic disease, it's okay. I think it's enhancement, then that's a whole different world.
Okay,
I agree with that too.
Yeah.
If your kid, if you want your kid to be strong, like you said, like that's some of the things like the muscle dystrophy. Like regardless of when it comes out and you didn't genetically modify it, it's not like they're not going to be like strong kids. You can have them go to the gym when they start getting older and stuff, and like if you want the kid to be smarter, like it's not about whether you should just genetically modify it to have it, or just like have it be born normally, and then wait to see if like that kid has problems or is healthy.
right. Anybody else have any? Yeah.
I'd like to agree strongly with that. The way we were talking about it is as though you just take a brick out and put another brick in, and then everything is going to be different. But we're not talking about the influence of environment. We're not talking about the influence of our willpower, of our minds, of our brains, and also the whole new area of epigenetics, which I was reading
Which
about.
is environmental.
Which is
Yeah.
environmental,
Yeah.
and so
Or
we're
some
not-
environment.
we're not understanding that there's all these other ways that can influence genetic modification.
Yeah, that feeds into the point, we don't know what we don't know, right? And so I think that's an excellent point. Epigenetics is a big deal too to think about. Yeah. Does anybody have any other final contributions to this? So I hope, yeah, go ahead.
So one more thing. My fear is that by focusing on genetic modification, gene editing, we're going to do less research on the causes, on the root causes of the diseases in the first place.
Well, actually, I think that goes back to here. So the way people understand the root cause of something in a lot of ways is to just take it out. So what I would do, for instance, is I studied a lot about how fats are made and stored inside of cells, and then I would relate the information that I found to the processes that are involved in, for instance, diabetes development, right? Because fat plays a really big role in that. And I would take yeast cells because we have a high homology, meaning that the pathways that make our fats, like a triglyceride or a cholesterol, is really similar in a yeast cell. And so working in yeast is sort of, for me, I'm not a fan of doing like mouse work or things that have like full-on nervous systems. So I always worked in cell culture. It's just more comfortable for me as a scientist. And so I would take these genes inside of these yeast cells and I would pop them out. And then I would say what happened, right? So doing genetic engineering and removing entire genes or changing the functionality of a gene is really valuable in understanding like the process of something. And that's one of the biggest ways, taking sort of a reductionist approach, is like if you remove this one thing, what happens? And it's so helpful in understanding the process. And then can you rescue it by putting that thing back in to test that exactly was the thing that caused this thing, right? So that's a valuable way to do it.
Do you have the sense that this use of technology will be more widely accepted by policymakers than stem cell research, which is in this country faced so many moral roadblocks?
So I think in this case and in this case, I mean, geez, who wouldn't want the opportunity to cure a cancer or like a viral infection, a deadly viral infection? I think that these would be absolutely embraced. Well, this is already happening, very widespread. Right, basically, this is the standard. The clinical studies are starting with this one, right? And I think the dicey conversation comes when you start here. Yeah. But these are certainly questions that we have to have nonetheless. Yeah.
And do you know of groups that are working to help inform policymakers?
Yeah, I think there are a lot of sort of bridge groups that sort of bridge the science to the government and policy that are certainly working very hard at that. I know a lot of my colleagues at Rockefeller are like on panels. They've been invited by national and international panels to provide their educated consult in these cases. But let me point out, when IVF was introduced in the 60s, it was met with a huge amount of resistance. It's not natural, we're playing God, the same types of arguments that we make here for either stem cell research or for, CRISPR use on germlines. And now, I mean, if you think about it, the statistics are pretty high in terms of how many women depend on IVF for their family planning. I know several of my friends, this is how they were able to, this is like commonplace. If you are not getting pregnant in some amount of time, you resort to IVF in many cases, and that's just normal, right? But years ago, when it was first introduced, it was not normal, and it was met with a huge amount of resistance. So I wonder, will this be another example of history repeating itself, where the use of CRISPRs on embryos, for instance, when you guys are older and have kids, will this be the thing that's just like your friends are all doing it, So, yeah, I don't know.
Thank you.
Cool, so thanks. Please feel free to get in touch at any time. My organization is basically, we just want to connect with people on science, and you're very welcome to get in touch with us at any time. We have a number of public lectures at our university. You can check our calendar, rockefeller.edu. And if you have any specific, more sort of K through 12 types of education questions, or if you want to get your school or group involved with what we do, please stay in touch. So it's all about social justice and making sure everybody has access so that we can make science a more inclusive situation. So thank you.